Slide 1. This slide tape presents a discussion of the cardiac pump, structure and mechanism. Slide 2. This unit presents 4 major topics. 1. The heart as a pump. 2. Function of the microanatomical component. 3. Molecular organization of the myofibril. 4. Mechanism of contraction. Slide 3. After you have completed this section, you should be able to fill in a chart naming the 2 phases of the pumping action of the heart, and for each phase state whether the muscle is relaxed or contracted, approximate ventricular volume, ventricular circumference, and volume of blood added or subtracted. Slide 4. The heart is a 2-phase pump. In phase 1, when the muscle is relaxed, the heart is in diastole. In phase 2, when the muscle is contracting, the heart is in systole. Slide 5. During diastole, when the muscular walls of the heart are relaxed, blood from the veins is added to the ventricular chambers. During systole, when the muscular walls contract, a portion of the blood contained in the ventricles is ejected. Slide 6. The pumping action of the heart depends upon contraction of the muscular walls of the ventricular chamber. In diastole, when the heart is relaxed, the left ventricular chamber holds approximately 150 milliliters of blood. Slide 7. In systole, when contraction is activated, the muscle cells within the ventricular wall shorten, and a portion of the blood within the chamber is expelled. In this example, 75 milliliters are ejected into the aorta. Slide 8. In the normal beat of a heart, about 50% of the blood in the ventricular chamber is ejected during systole. Slide 9. The decrease in ventricular volume is caused by a decrease in ventricular circumference. The circumference in this example is 21.1 centimeters at the end of diastole and 16.1 centimeters at the end of systole. Slide 10. The decrease in circumference is due to a shortening of each muscle cell or element in the circumference. Slide 11. Although in this model the ventricular wall is oversimplified, the principle we want to get across is that during systole it is the shortening of each muscle cell which causes circumferential shortening and hence ejection of blood. Slide 12. Following systolic ejection, the ventricular muscle relaxes. The phase of diastole is initiated, and the chamber is refilled with blood. The energy needed to fill the ventricular chambers is provided by the pressure of blood in the atria and veins. The pressure of the blood in these structures determines the volume to which the ventricle fills. Slide 13. To assess your understanding of this section, complete this chart. Stop the tape while you work. Slide 14. Slide 15. The objective of the next section is to enable you to select a function from a given list to match each microanatomical structure. Slide 16. In this section, we will be interested in the structure of the muscle machine and the mechanism by which a source of energy is used by this structure to change cardiac muscle length and hence pump blood. Although we do not know in complete detail the structure of the machine or exactly how shortening takes place, much is known about both problems. Slide 17. Since our theories concerning the initiation of systole, contraction, and the termination of systole, diastole, as well as the mechanism of shortening, are closely allied to what we know about the structure of the cardiac muscle cell and the contractile element contained therein, we will start by describing the microanatomical structure of the cardiac muscle cell. This slide provides a view of the heart at successively greater magnifications. The heart and great vessels are shown with a portion of the right ventricle cut open. This cut portion is enlarged and seen to be composed of many cardiac muscle cells. Slide 18. The cells are separated from each other by an intercalated disc and by the sarcolemma, which is the limiting membrane of the cell. Slide 19. The sarcolemma invaginates the muscle cell at regular intervals with a structure called a T-tubule. Both the sarcolemma and T-tubules generate action potentials. The cardiac cells contain numerous mitochondria. These organelles generate the ATP, which is used to fuel the contractile apparatus. Slide 20. Abutted against the T-tubule is a complex tubular network, the sarcoplasmic reticulum. The sarcoplasmic reticulum, which surrounds the contractile elements, the fibrils or myofibrils, acts as a sink and perhaps a source of calcium for the myofibrils and is involved in initiating and terminating contraction. Slide 21. This portion of one of the cells identifies the myofibril. The myofibril is the contractile element within the cardiac muscle cell. It is the shortening of the myofibril which causes the cardiac muscle cell to shorten. Slide 22. Here we see the heart from organ to myofibril. Slide 23. Now check your knowledge of the structure and function of a muscle cell. Stop the tape while you answer. Slide 24. Slide 25. After completing this section, you should be able to label diagrams of the following myofilament structures. 1. Sarcomere with thick and thin filaments. 2. Thick filament with an assembly of myosin molecules and their cross bridges. 3. Thin filament with the polymerized G-actins and associated control proteins, tropomyosin and troponin. Slide 26. The myofibril, as shown by electron microscopy, is composed of repeating units called sarcomeres. Each sarcomere is bounded by 2 Z-lines. Slide 27. The sarcomeres, in turn, are made up of arrays of 2 types of structures called thick and thin filaments. The geometric alignment of these filaments is shown in 2 dimensions here, slide 28, and 3 dimensions on this slide. Note that each thin filament is located equidistant from 3 thick filaments. Slide 29. The thick filament is assembled from a large number of myosin molecules. Slide 30. The myosin molecule has a molecular weight of about 490,000 and is composed of 2 alpha helical strands combined in a supercoil. Each strand terminates in a globular head. The 2 globular heads contain ATPase, the enzyme needed to release the energy contained in ATP. Slide 31. The backbone of the thick filament is formed from the alpha helical portions of the myosin molecules. Since the myosin molecules are arranged in a tail-to-tail fashion within the backbone of the thick filament is polarized with symmetry about the M line. Slide 32. This arrangement of the myosin molecules in the thick filament may order the direction of contraction toward the M line. The myosin heads, which contain the enzymatic portions of the molecules, stick out from the backbone of the thick filaments and form structures called cross bridges. Slide 33. The major structural component of the thin filament is G-actin, which is polymerized as a double helix. In addition, the thin filament contains tropomyosin and a complex of troponin molecules, troponin I, troponin T, and troponin C. Slide 34. The tropomyosin molecules lie end-to-end along the grooves of the actin helix and are involved in regulating the attachment of cross bridges to the thin filaments. Slide 35. At about 130 angstrom units from the end of the tropomyosin molecule, the troponin complex is attached to the thin filament. Slide 36. Tropomyosin plus the troponins constitute the regulatory proteins. The presence of the regulatory proteins on the thick filament prevents interaction between the thick and thin filaments, except when calcium is bound to one of the troponin molecules, troponin C. Slide 37. Calcium binding to one of the regulatory proteins, troponin C, allows the thick filament to interact with the thin filament. Slide 38. To determine your understanding of the structure of the myofilament, label the three following diagrams. Stop the tape when necessary. Slide 39. Slide 40. Slide 41. Slide 42. Slide 43. Slide 44. The last section will discuss the mechanism of contraction. After completing it, you should be able to diagram the following processes. 1. Myofilament translocation. 2. Action potential activation. 3. Thick and thin filament interaction. 4. ATP involvement. Slide 45. Examination has been made of filament length during changes in sarcomere length induced by passive or active mechanisms. Careful measurement has shown that filament lengths do not change during changes in sarcomere length. Slide 46. It has therefore been concluded that sarcomere shortening takes place by a sliding mechanism, that is, by the translocation of thin filaments past thick filaments. During shortening, the thin filaments move toward the middle of the thick filaments, that is, toward the M line. Slide 47. The return of the sarcomere to the length from which shortening began is thought to be a passive process. Slide 48. As mentioned earlier, The energy for lengthening is provided mostly by the pressure of the blood in the atria and veins and takes place when thick and thin filaments are not interacting. Metabolic energy is thus not required to return the sarcomeres to their initial length. Slide 49. Sarcomere shortening, however, requires filament interaction and a source of energy. The immediate source of energy for filament interaction is provided by the hydrolysis of the terminal phosphate of the ATP molecule. Slide 50. The mechanism by which sarcomere shortening progresses is not known for certain. There is, however, a rich body of evidence indicating that it progresses as a consequence of interaction between the thin filament, calcium, magnesium ATP, and the thick filament. Slide 51. The sequence of steps involved in cardiac activation and sarcomere shortening is shown on this and the next two slides. In step 1, the cardiac action potential releases calcium. Calcium binding to troponin C in step 2 produces activated thin filament sites. Slide 52. The activated sites allow myosin heads, that is, cross bridges, to attach to thin filaments as shown in step 3. The attachment of a cross bridge to a thin filament allows a small rotation, step 4, of the cross bridge and a translocation of the thin filament relative to the thick filament. Slide 53. Following rotation, the cross bridge detaches from the thin filament, step 5. Cross bridge detachment allows the thick filament to interact with another activated thin filament site. This yields an unattached cross bridge, step 6, which after appropriate activation can attach to a new site on a thin filament. The entire sequence is shown on this slide. Slide 54. The microanatomical correlates of the contraction cycle, steps 3, 4, 5, and 6, are shown on this slide. An activated thin filament is shown attaching to an activated thick filament with the subsequent rotation of the cross bridge and translocation of the thin filament. This is followed by detachment of the cross bridge and the formation of a new activated cross bridge. Slide 55. In order to achieve macroscopic shortening of the type required to eject blood from the ventricle, the cycle of steps 3, 4, 5, and 6 is repeated many times. The energy required to drive this cycle is provided by the hydrolysis of ATP. The reaction sequence outlined in the previous slide leaves unspecified the connection between ATP hydrolysis and contraction. The connection as it is now understood is outlined in the next few slides. Slide 56. ATP enters the cycle after cross-bridge rotation, step 5, and is responsible for cross-bridge detachment. The bonding of ATP to the cross bridge causes it to detach from the thin filament. Slide 57. Following ATP binding and cross bridge detachment, step 6, myosin ATP decomposes to a complex of myosin, ADP, and P, the activated cross bridge. Slide 58. The myosin ADP-P complex next attaches to an actin site on the thin filament to form an actomyosin complex, step 3. Slide 59. This highly energetic complex produces rotation of the cross bridge and translocation of thick and thin filament, step 4. Slide 60. Following translocation, ADP and P dissociate from the actomyosin. ATP enters again dissociates the actomyosin complex, and the cycle is repeated. The entire cycle is shown on this slide. Slide 61. In order to produce muscle shortening of a sufficient magnitude to eject blood from the ventricle, this cycle must be repeated many times. Thus, many molecules of ATP are hydrolyzed with each beat of the heart. The resynthesis of this expended ATP requires oxygen, which must be supplied to the mitochondria. These facts are of particular importance when one approaches the treatment of those heart diseases where the availability of oxygen is compromised. Slide 62. The next three questions assess your understanding of the processes involved in the mechanism of contraction. Stop the tape to draw diagrams when necessary. Slide 63. Slide 64. Now diagram activation of the contraction mechanism. Slide 65. Slide 66. Diagram the process of thin filament translocation. Slide 67. Slide 68. Finally, show ATP involvement in the contraction cycle. Slide 69. Slide 70. Now that you have completed this slide tape, you should review the topics which have been presented and take the post-test at the end of the study guide. Slide 71.